I have a number of slides. We're going to get going. The first half of the presentation, I'll give some breeding updates at the University of Minnesota. Second, if I'll talk about this pollen dispersal study, it's out of my comfort zone, but I thought that was a cool study that I should share instead of talking about genomic selection and mapping all the time. Here's a brief outline. I'll show you some cycle 5 data, which is our current breeding cycle. It'll end this fall and we'll start cycle 6 this fall, and talk about how I selected individuals for the cycle 6 population. Touch briefly on variety candidates performance. I have 1 year data, show some yield and seed size information there. And a root study, someone was asking, I think Jessica, about what's happening in the breeding conditions. I'll talk about and majority of the talk will focus on pollen dispersal in wheatgrass. So here's how it at the university. We have 600 to 700 space plants that are about 3 feet apart, slightly less than a meter. This selection nursery is also our genomic selection training population. There's about 65 to 75 family cycle with about 8 to 9 half-sibs, meaning siblings within the family. We plant this in 2 locations within Minnesota, St. Paul, which is where we had the campus, and Lamberton, slightly southwest of Twin Cities. And they're mostly evaluated for 2 years, occasionally a 3rd year. You saw traits already in previous slides, not gonna go through all of this, but I will highlight lodging resistance. Minnesota gets a decent amount of rainfall, strong winds in the summer, so you keep an eye on that. I do select for resistance to scab and ergot. And end-use quality is something the breeders don't directly, well, I personally don't directly work with, but we collaborate with food scientists on campus to get information on how our variety candidates are doing. Here's a look at our cycle 5 performance. These are space plants again. 1 year data from last year. Maroon is Lamberton, gold is St. Paul. The first thing you'll notice is that Lamberton means are less than St. Paul. That's normal, it happens, it changes between locations. But the key thing here is that the difference we see, which is called, in short, genotype by environment interaction effect, that has helped us improve our genomic selection. It actually boosts the prediction. So we use that data to select our future populations. And a quick overview of how we do it. We'll start with the training population right here, top at the top there. That's again our space plant nursery. Once you have the phenotypic data and genotypic data, which is sequenced GBS, you might have heard of it. We train our genomic selection models. We use that G/E information and a breeding population, which is essentially the progeny of the best individuals in the training population, are only sequenced. They never see the field, about 3,000 to 4,000 seedlings. Those go through the same models. And from that, I pick 70 to 100 best individuals that are intercrossed in the greenhouse, and it forms the new cycle and so on. The right-hand side, I don't have time to go through it today, but you can see that anything we do on the left side, any decision we make, the genomic selection model selection, it does affect how we develop our variety candidates. Here's a look at the predictions. So first of all, again, to reiterate, the model used the G/E information. The trait data was corrected for spatial variation using ASREMO. And 10,000 iterations, 100 reps using 4-fold cross-validation method. On the x-axis on the plot, I have traits. On the y-axis, I have predictability, which essentially is correlation between your traits and field observed traits. The numbers vary for traits. This is very normal in wheatgrass, in other species as well. Some traits have high means, others have low. Of these, I personally put the most weight on 1,000 kernel weight, seed width, and threshability. Those are important traits, but not too far behind are shattering and grain yield. Those are, I would say, are my top 5. Other traits such as plant height and spikelet numbers and so on, I keep an eye on it, but they're not, I don't focus on them too much. So based on the predictions, I selected about 100 individuals. Of those, about 70 or so are being crossed in the greenhouse as we speak right now. The progeny would be planted, cloned, and planted in the field some near the end of August, and that will form our cycle 6 population. Moving on to variety candidates, we had some candidates in 3 Minnesota locations: Lamberton, St. Paul, and Roseau. Roseau is pretty much at the extreme north of Minnesota, close to Canada. If you look at the yield data on the chart towards the right, the data is relative to MN Clearwater. So if MN Clearwater is 100, which is right around here, MN 1603 was about 11% better than MN Clearwater. TLI 701 and 703 did pretty well. 704 was the best overall out of all locations, slightly over 1,100 pounds an acre in Roseau, which is pretty impressive. MN 1603 is also one of the variety candidates being used by all the KernzaCon participants. So very excited to see how it does in these multi-sit trials. Here's a look at seed mass or 1,000 kernel weight, whatever you want to say. MN Clearwater is near the end at the very edge of the chart here. 1603, again, it was the best of all. It had about 17% larger grain compared to MN Clearwater. So like Lee said, it's very exciting that these new varieties are performing better. Anything we'll be doing in the future, I hope we can continue this progress. And to reiterate another thing Lee said, this is only 1-year data. We need at least 3-year data, in my opinion, to make a decision on which candidate will be released. And at least for the University of Minnesota, the next variety release will likely happen near the end of 2023 once we have 3-year data. This is my slide on the breeding portion. So here's a schematic of the root study that we're going to initiate this fall with collaboration with Dr. Jake Youngers. So far, there's been no root study in wheatgrass breeding populations. We don't really know what happens in our space plants under the soil. So we're going to put these mini rhizotrons, as you can see in the cartoon. In the cartoon, it's shown in an angle. The current protocol from Dr. Youngers, it goes vertically because of logistical reasons. But we're going to do this in about 150 randomly selected plants. If I have genotypic data before this fall, we can maybe even select them based on genotypic differences. And we'll be measuring root data starting this fall and next year twice at least. And I hope to continue this project beyond that, depending on funding. We'll collect different root properties such as number, length, diameter, and area density using machine learning methods. And we'll do this at 4 depth, a depth of 100 centimeters. The trait will be related with above ground data to see what's the relationship between underground root information and above ground trait data that the breeders mostly focus on. Now, I know that 150 is a small sample size. Nonetheless, I hope there's an opportunity to expansion in the future to a larger size so our models can be better. And of course, based on both above and underground information, we'll select the best individuals to be put in crossing blocks so that our future variety candidates have both above and well, better both above and underground information. So again, very excited to initiate this project this fall. So if I'm gonna move on to pollen dispersal, I'm glad the video worked. I was not sure it was gonna work. So most of us know that wheatgrass is predominantly wind pollinated species. There's limited insects and pollinators pollinating, but it's majority of it is wind. You can see plants swaying and rocking in the background. That's how the pollens get distributed across the field. And they share genetic information intermixing. This is how we maintain diversity. This is essentially how we can carry out our selections because of the gene transfers that's happening. If we look on cartoon level, consider these as 2 individuals where there's poles from individual 1 on the left to the right and from right to the left, because in nearly all cases, mothers, fathers, and fathers, mothers in wheatgrass. Obviously, we don't have only 2 individuals. We have hundreds of spaced plants where the pollen is moving freely from one to other all across the field. And it's pretty cool that there's intermixing going on, and this intermixing of genes and alleles that are shared in the population is essentially what is is a measure of gene flow, and that in turn determines population structure, diversity, and like I said, it helps maintain the diversity long term. And to illustrate my point, if you look in the figure on the right, if we start at the very bottom and consider that to be the Rodale Institute population, for example, and somewhere near the middle where you can see the T, the dashed line is TLI cycle 3. TLI cycle 3 is the population that we gained from the Land Institute to start our breeding program at Minnesota. So then the two branches above there, if you consider either one to be University of Minnesota population and the other one to be the Land Institute, you can see there's these spurs or these veins that are flowing. While they were at initially around the same stage, they've changed as you move to the very top, and that's because TLI is selecting plants that adapt the best to Kansas environments, and we are doing the same that adapt the best in Minnesota environments. So as we do that, there's shift in genetic diversity, gender of populations, and while the cartoons on M2, these are alleles, basically intermixing between the two populations going on. We haven't done quite actively yet, but that does present an opportunity to even improve it in the future. But my point is that this gene happens in an initial population. It tends to get separated out and diversified as we select our populations for a specific purpose. And speaking of selection, we know that a strong selection, either phenotypic or genomic, genetic diversity. And as we continue to make selection, make progress, we've got to kind of keep an eye on what's our diversity levels in our populations. Differential pollination or differential flow, which basically means maybe a bunch of flower together, or there's pollination among fields that are far apart. That could also impact genetic diversity. This is something to keep an eye on. And the articles on the right, excuse me, kind of they support these statements. And at the same time, if a population experiences broader dispersal, which, like I said, if there's a crossing between individuals from TLI and UMN, that fosters diversity. It's all about diversity, mainly because we want to keep that, maintain that, promote that, so we have better individuals, more individuals to select from in our populations currently and in the future. So with all that said, we conduct this pollen dispersal study in wheatgrass with the main objective of understanding what is the extent of pollen dispersal. We know it disperses freely, it's mating, but like what exactly are the numbers in our fields? We specifically studied parameters and patterns of mating in wheatgrass. We identified parentage of the progeny that we use to initiate our next cycles. We also looked at patterns of genetic diversity in both parents and progeny, and then finally looked at how pollen dispersal or this gene flow through distance, it affects progeny diversity and trait performance. And I must thank PepsiCo for making this project happen, because they gave us the decent amount, and we were able to sequence a bunch of tools with the money they gave us. Here's the setup for this study. We use cycle 4 population. The figure on the right is a busy figure, but the blocks, the dotted rectangular blocks, each block is one family. So within each block, there are 9 half-sibs. On the very right, there are no blocks. They came from other biparental crossings, but because they're in the same field, they were included in the study, but they're not half-sibs relative to the main population on the left side. Overall, we sampled about 846 individuals or progeny from 50 mother plants. You can see the mother plants in those black circles all across the field. I tried to pick them in random order, yet kind of inside the fields, so that they would have a decent amount of pollen load around them. And we had a mean of 56 progeny per mother plant. They were sequenced using GBS, and I had about 21,000 markers at 20% missing data, which is it's fairly normal in wheatgrass. First thing we did was we found the fathers of those sampled progeny, because again, we know the mothers, we don't know the fathers, and we use a program called SERV to do so, and that program identified fathers of 91%, excuse me, of me. And we observed that for each mother plant, there were 19 to 47 fathers contributing pollen, on an average 37. And the average distance between a mother and a pollinating father was 7 meters, which is roughly 8 plants in our field. And I guess the biggest discovery from this study was that 52 fathers were within 5 meters and 80% were within 10 meters. So when you are out in the field tomorrow, if you walk about 5 meters radius and then go in a circle, you can say that 80% of the pollen to that central plant comes within that level. And that's pretty cool. The figure on the right illustrates what I just said: distance, or pollination distance on x-axis, and number of progeny on the y-axis. We're going to look at the same plot, but through the perspective of number of fathers. The colors show which mothers the fathers pollinated. And on the y-axis, number of fathers; on the x-axis, the distance bin. You see 0 to 3, 3 to 6. That's arbitrary. I could have said 0 to 5, 5 to 10. It doesn't matter. But the point is that as you move away from the mother plant, the number of fathers reduces. And as I mentioned, 80% of the fathers come within 10 meters of the mother plant, and the rectangular blocks illustrate that. I looked at the effect of trait attribute of the fathers in pollination success. Heading: if a plant emerged, it spikes earlier than others, does that help in pollination? If a plant is taller or it has higher spring growth vigor, and if adds towards more fertility, they have any impact on pollination? And the answer was no. So being tall or the spikes out early really help that father become more successful in pollinating the mother plants. And that's actually a good thing, because it means that while the plants, majority of them are within a short distance of the mother plant, you can still expect randomness in terms of where the mother plant is getting its pollen from. So that was something pretty cool. Going back to the half-sibs, because we have a family structure-based space plant nursery, we looked at their success rate in pollination. And if you look at the plot, and again, distance on the x-axis and proportion of fathers on the y-axis, the blue line is half-sibs, meaning that if there was a mother plant within a family, the blue line shows the success rate of the half-sibs within that family in pollinating the mother plant from the same family, whereas red line is non-half-sibs, fathers outside that family. So overall, except the very first scenario, overall in the nursery, found out that the half-sib fathers are actually more successful than any other given father in the population. And that was very interesting, because I personally had thought that maybe if individuals are coming from the same family, there might be some incompatibility issues, but that was not the case. We did not track down SI alleles and their composition in the families, but overall, there was no drawback towards having half-sibs planted side by side. We do that in our nursery because we can see how a family is doing together. As I'm walking in the field, I can just compare side by side instead of having to, spot down the half-sib if it was planted randomly elsewhere in the field. So that was a confirmation to our method that it doesn't really matter as long as, it does not matter if the half-sibs are randomly planted or not. I mentioned about genomic selection earlier. If you look at the breeding population portion here on the top left, that our progeny we sampled for this study is a representation of that breeding population. So anything we learn on this progeny can be then applied to our actual breeding population that we use in genomic selection. And for that, what we did was we first looked at the trait performance of the progeny as it comes out or predicted by the models, and looked across different distance bins. So again, on the x-axis, there is pollination distance. On the y-axis, there's predicted phenotypic value from the genomic selection models. And overall, there was no significant differences. There were a couple instances where the very first bin and the very last two bins were significantly different. I'll mention that why in the next slide. So we looked at diversity of the progeny. I used 4 metrics. There are different metrics, kind of similar, but I wanted to look at different metrics just to be sure that we are not missing out on measuring the diversity in the population. And here as well, Across the distance bins, we did not see significant differences, except in certain cases at the very last 2 distance bins, you can see the numbers fluctuate quite a bit. And that's because the last 2 bins, I think, had less than 10 or 12 individuals. So that's because of a sample size issue and not necessarily a true attribute of what the plants look like. So all that said, what is the summary of this pollination study, and why do we care? Well, Steve, if you go back to Logan and you find out that our field has burned except that small section, you'll be okay, because when you sample progeny, if you have to start out fresh from just the samples in that block, you can still expect to have enough diversity and good enough trait performance. And this is not to say that you will not lose an individual with the best or the largest seed that's on the edge of the field, or the one that produces the most grain, but there should be enough diversity in the population you sample from that middle area to start fresh and catch up in maybe a few cycles. So takeaways, we found a vast majority of the pollination happens within 10 meters. And the trait performance of others had no apparent effect on pollination success. Half-sibs were also more successful than non-half-sibs in pollinating the mother plants within the family, and neither progeny performance, predicted performance by genomic selection models, nor their diversity was affected by the pollination distance bins, which is again the distance of fathers. And the biggest takeaway is that our current method of using genomic selection and selecting progeny the way we do, I think we can go on doing that for quite a while, because it has no apparent effect on reducing genetic diversity. So with that, I'd like to end by acknowledging a bunch of people, a lot of support, a lot of help within and outside of Minnesota. Our funding mainly comes from Minnesota Department of Ag through the Forever Green Initiative. General Mills supported us for a while. FFAR is supporting us currently. And some of the support also comes from the KernzaCap through USDA and EFI, which you heard about. So if I have any time, I can take questions. If not, we can catch up later. Thank you.
We have time for questions.
Yes, Steve.
Do you have any, has anyone looked to see, like in these synthetic varieties where you have as few as 5 parents, how equal are those parents contributing to those synthetic varieties? And then a second part to this question, which both you and Lee is if you think that you could, when you start doing these yield trials of these candidate varieties, the synthetic varieties, you think you could make a genomic selection model for yield plots based on the genotypes of the parents?
So I can start with the first question, which was, have you done similar study or looked at synthetic populations where you use 5 individuals or 10 individuals? That's a great question. And I am currently actually evaluating that. MN Clearwater came from different number of parents. We have currently MN1603 came from just 4 fathers, 4 parents. Our newest candidates have 10 parents. So I do have different combinations, and I'm currently evaluating using simulation and real data to see how the diversity changes as you move from scene 1 to 2, 3, 4, 5. We don't have real performance on all of those. We do have some on MN Clearwater for 3 years. But that's in a study that's happening currently, and I hope I'll have some answers maybe by fall or early winter this year. And the second question was genomic prediction of yield of synthetics using fathers. I think it potentially could be done. I have personally not done that. I'm not sure if Lee has. I think selected the individuals at least based on some genomic index stuff, but the half-sib projects we're currently evaluating that could lead into that, because we can use the data from the mother plants and predict maybe the half-sibs. But so far we have not done that, and that's an interesting avenue for research.
More questions? Yes, okay.
I don't know if you're going to know the answer to this. It might be a Jake question, but the mini rhizotrons that you're talking about in the root study, how long does it take to sample one of those? Because you said 150 samples, that's not that much. I think 150 mini rhizotrons to check, that's kind of a lot.
Yeah, breeders always want more and more. I'd prefer 250, obviously, but we have limitations. I need to respect Jake's time. 48 samples take 8 hours. 48 plants take 8 hours, so a full day. So to evaluate 150, it'll take us 3 days, 4 days probably. Valentina had a question?
Yeah, do you have plans for like a variety trial, multi-site? Are you testing it in multiple locations? And is there any coordination in the Kansas, Minnesota on that regard?
Yeah, actually I think there is. I try to send some of my best stuff to Lee. Lee sends his best stuff to me all the time. You know, you saw TLI 701, 702, and we're going to put new yield trials this fall, so I'll be sending new stuff to Lee and he'll send his new materials to me. But yeah, there is the coordination and collaboration there for sure. And again, to highlight the work KernzaCap is doing, I think that's going to be huge. That's evaluating all these candidates. And many of those have not been released. MN1603 looks like the strongest candidate to be released. So I'm very excited to look at the data this year and next year.
All right, thanks, Prabin.
All right, thanks all.
Great.